Welcome back to this next video, and this is the part 2 on the G-alpha Q signaling through the IP3-DAG pathway, and we were discussing about the activation of the protein kinase C as an example of the serine threonine kinases. A quick reminder of what we discussed in our first video is that when the ligand it binds to the G protein coupled receptor, the alpha subunit move from the GDP into the GTP form, and the alpha GTP then goes and activate the phospholipase C, which actually is responsible for the breakdown of the PIP2 into the IP3 and DAG, and this IP3 was then going to bind to the endoplasmic reticulum to release the calcium into the cytoplasm, and then that particular calcium, and that we will discuss today, that how the calcium and this DAG is then actually responsible for the activation of the protein kinase C. We generally discussed about the structure of the G protein coupled receptor, that is a 7 transmembrane protein. It means that it is spanning the membrane 7 times, creating the extracellular loops for the binding of the ligand and the intracellular loops for the binding of the heterotrimeric G protein. We discussed about the different type of the alpha subunits, that there are alpha subunits which usually belong to 4 different types. One type of the alpha subunit was the G alpha I, the other one was G alpha S. The one that we were focusing on was the G alpha Q subunit, and there are other types of the alpha subunit which are known as the G alpha 12/13. I told you that when the alpha subunit it gets a GTP after the binding of the ligand to the G protein coupled receptor, it actually activates the phospholipase C beta enzyme. Excuse me, and this phospholipase C beta enzyme was responsible for the breakdown of this phosphatidyl inositol 4,5 bisphosphate, the structure of which we discussed in detail in the previous video. And when the phospholipase C it acts on the PIP2, it is actually broken down into the diacyl glycerol and the inositol 1,4,5 trisphosphate, which are the IP3. And the IP3 then was then going to bind to the inositol trisphosphate receptor, which was present on the surface of the endoplasmic reticulum, thereby allowing the release of the calcium into the cytoplasm because of the concentration gradient. Then we discussed in detail about the calcium homeostasis. We discussed about the SERCA, the PMCA. Then we talked about the SOCE channels. After the stem 1 and the stem 2, they oligomerize and they bind over there to replenish the stores of calcium in the endoplasmic reticulum. In this particular video, I want to focus on the protein kinase C, for short, which are known as the PKC, which is the protein kinase and then C. Now, these protein kinase C, they are actually serine threonine kinases. What I mean by that is that when they act on their substrate, they are going to phosphorylate them. Therefore, they are known as the kinases. And the protein kinase C is a special kinase which is known as the serine threonine kinase. What it means is that when the protein kinase C it acts on its substrate, it is going to phosphorylate its serine or threonine residues. So the protein kinase C, they are actually activated by signals such as increase in the concentration of the diacyl glycerol or calcium. That we just saw that because of the binding of the ligand, the phospholipase C is going to make the DAG, and the IP3, and the IP3 was then going for the for increasing the concentration of the calcium ion in the cytoplasm of the cell. Now, the protein kinase C family, it consists of 15 isozymes in humans, and they are broadly divided into 3 major families, and these families, they are based on their second messenger requirement. Each family required a different second messenger for their activation. The first class of the protein kinase C, they are known as the conventional PKCs, and if you talk about the members of this family, the members of the conventional PKC family include the alpha, the beta 1, the beta 2, beta 3, beta 4, and gamma. So these are the members of the protein kinase C which belong to this conventional protein kinase C family. Now, if you talk about the activation of the protein kinase C in this conventional family, they require calcium, the diacylglycerol, and a phospholipid such as phosphatidylserine for their activation. It means that if you want to convert them from their inactive to active state, you will be requiring these three things: the calcium, the diacylglycerol, and a phospholipid, for example the phosphatidylserine. The second family of the protein kinase C, they are known as the novel PKCs, and the member in this particular family include the delta, the epsilon, the eta, and theta. These are actually the members in this particular family. Now the protein kinase C in the novel protein kinase C family, they do require the diacylglycerol for their activation, but they do not require calcium for their activation. What it means is that the activation of the conventional and the novel PKCs, they are activated to the same signal transduction pathway as the phospholipase C, because this DAG is required in both of the family, and this DAG is actually made by the action of the phospholipase C on the PIP2. Another family of the protein kinase C, that is known as the atypical protein kinase C. Atypical refers to something that is unusual. Therefore, because these protein kinase C in the atypical family, for example the zeta protein kinase C, they neither require calcium nor diacylglycerol for their activation, but they require a different second messenger, which is presumably generated through a phosphoinositide or the PI3 kinase pathway, because this the zeta protein kinase C, for example, it do not require neither the calcium or the diacylglycerol. Therefore they are known as the atypical protein kinase C. But collectively the conventional PKCs, the novel PKCs, and the atypical PKCs, they are making the family of the protein kinase C. Now if you look at the structure of the protein kinase C, there are specialized domains in the structure of the protein kinase C. On the N-terminal of the protein kinase C, there is a pseudo substrate domain, and we will discuss its function in a while. Then you have got the regulatory domain, and under the regulatory domain you have got three things. Under the regulatory domain, you have got this pseudo substrate domain over here. You have got the C1 domain, and this C1 domain is actually the DAG binding site. You have got this C2 domain, which is actually responsible for binding to calcium, and then you have got these catalytic domains, and there are two catalytic domains. One is known as the C3, which is responsible for the binding of the ATP, and if you talk about this C4, that is actually the substrate binding site. So this is actually the structure of the conventional PKCs. For short, you put this C over here just to represent that this is conventional, and then the PKC. So on the N-terminal, you have got this regulatory domain containing the pseudo substrate, the C1 and the C2 domain. And then you have got two catalytic domains, the C3 and the C4, which are responsible for the binding of the ATP and the substrate binding site. Now this C1 domain is further divided into two subdomains. One is known as the C1a, and the other one is known as the C1b. But collectively, the C1a and C1b, that is responsible for binding to the diacylglycerol that we will see in a while. Now, if you talk about the novel PKCs, as you can see over here, there is no calcium binding site over here because they do not require the calcium for their activation. Now, I'm not going into the detail of the atypical protein kinase C because they require different second messengers for their activation. But the structure of the conventional and the novel PKCs, that usually differ by the presence of this calcium binding domain in the conventional PKCs and the absence of this calcium binding domain in the novel PKCs. Now, if you talk about the inactive form of the conventional PKCs, what happens is that when there is no signal and you do not need the activity of the protein kinase C, what the pseudo substrate domain do, I'll represent the pseudo substrate by this PS. This P is for pseudo and this S for the substrate. So during the inactive form of the conventional PKCs, the pseudo-substrate domain, it goes and it binds to the substrate binding site, because the structure of the pseudo-substrate, it have got similarity with the substrate of the protein kinase C. Therefore, this pseudo-substrate binding site, it goes and it binds to its substrate. So when it binds to its substrate, that means that the substrate binding site of the protein kinase C has been occupied, and it will not be able to interact with its substrate, and therefore it will not be able to phosphorylate them. So when the pseudo substrate site, it goes and it binds to the substrate binding or the C4 domain, it renders the protein kinase C in the inactive form. Now, when you want to activate them, what happens is that when the concentration of the calcium that increases because of the binding of the IP3 to the IP3 receptor on the endoplasmic reticulum, the calcium is moving from the endoplasmic reticulum into the cytoplasm, increasing the concentration of the calcium. Now, when the concentration of the calcium that increases, the calcium, it actually goes and it binds to the C2 domain, which was actually the calcium binding domain. And to this calcium binding domain, two calcium ions, they go and they bind to the calcium binding domain. So when the calcium binding domain that have been attached with the two calcium ion, what happens is that the pseudo substrate site, it will be actually removed from the substrate binding site because of the binding of this calcium. So you will be actually having a change in the conformation of the protein kinase C, because initially, as I've told you, that in the inactive form, the pseudo substrate site, it is going to bind to the substrate site. So when there is calcium on the C2, the pseudo substrate, now it do not have the ability to bind to the substrate binding site. This means that the substrate binding site and hence the catalytic domain of the protein kinase C, that will be free to act. Now, when the calcium, it binds to the calcium binding site, there is a special protein in the cytoplasm, which is known as the RAC protein. Now, this RAC is actually for a receptor for activated C kinase. What I mean by this RAC protein is that when the calcium, it binds to the calcium binding site, then it is going to interact with this RAC protein, and the RAC protein will translocate the protein kinase C from the cytoplasm into the cell membrane. Keep in mind one thing, that the protein kinase C is not a membrane bound protein, it is there in the cytoplasm. So when the calcium, it binds to the calcium binding site, this particular form of the protein kinase C then interact with the RAC protein, and the RAC protein actually translocate the protein kinase C from the cytoplasm towards the cell membrane. Now, in the cell membrane, the DAG site, it actually interacts with the diacylglycerol. As I've told you, that this particular C1 domain containing the C1A and the C1B domain, they are actually responsible for the binding of the DAG. So if the protein kinase C has to interact with the DAG, it must move from the cytoplasm towards the cell membrane, and the RAC protein is actually helping the movement of the protein kinase C from the cytoplasm into the cell membrane. So in the cell membrane, the protein kinase C is then going to interact with the diacylglycerol and another component which is known as the phosphatidylserine. And when the protein kinase C interacts with the calcium, the diacylglycerol, and then the phosphatidylserine, the conventional PKCs that actually gets activated. What I mean by that is, when they get activated, they can actually phosphorylate their substrates now, because the substrate binding site is now free for the substrate binding. Now, when the protein kinase C, it gets activated because of the binding of the calcium, the DAG, and the phosphatidylserine, the protein kinase C is then going to phosphorylate specific serine or threonine residues on the target protein. Now, that vary depending on the cell type. Now, if you talk about the highest concentration of the protein kinase C, they are actually found in brain, and where among other things it phosphorylates ion channels in the nerve cells, and when it phosphorylates the ion channel at the serine and threonine residues, it actually changes their properties and altering the excitability of the nerve plasma membrane, which is actually responsible for the generation of the action potential, and we will discuss that in detail when we discussed about the neuronal signaling. But for this particular time, the highest concentration of the protein kinase C is found in the brain, and when it gets activated by the calcium, the DAG, and the phosphatidyl serine, it goes and it phosphorylates the serine and threonine residues of the ion channel in the nerve cell, thereby changing the excitability of the nerve plasma membrane. Now, if you remember when we were discussing about the homeostasis of the calcium, I've told you that some of the calcium, it actually binds to a cytoplasmic protein, which was the calmodulin. So now we are going to have a detailed discussion on this calmodulin. Now this calmodulin, it is a ubiquitous intracellular receptor of calcium. It is present in the cytoplasm, and it is actually acting as an intracellular receptor of calcium. Now this calmodulin, this is a calcium binding protein, and it is found in all eukaryotic cells. And if you talk about its number, why it is called is the ubiquitous. So a typical cell contains more than 10 raised to the power 7 molecules of calmodulin, which is a very huge quantity. Now this calmodulin is a highly conserved single polypeptide chain made up of about 150 amino acids, and it has the ability, a single calmodulin polypeptide has the ability to bind to 4 calcium ions. It means that the single polypeptide of the calmodulin have got 4 high affinity calcium binding sites. And when the calcium it binds to it, the calmodulin actually goes a conformational change, and when it goes a conformational change, then it is going to interact with its substrate that we will discuss in a while. Now the calmodulin, it actually have got 2 globular domains, and these 2 globular domains are connected to each other by a flexible central linker. In each domain, each of these 2 globular domains have got 2 calcium binding sites. It means that there are 2 globular domains. To each globular domain, 2 calcium ions they are binding. So a total of 4 calcium ions they actually bind to a single calmodulin polypeptide. Now the calcium binding sites in these globular domains, they are actually made up of 12 amino acids. And these 12 amino acids usually contain negatively charged or polar amino acid residues, such as the aspartate, the glutamate, and the asparagine. Now, if you talk about the calcium, it have got a positive charge. If you talk about the negatively charged amino acid, they have got negative charge. So the negative and the positive, they are actually going to interact with each other. So the calcium is then going to interact with the calmodulin, utilizing the aspartate, the glutamate, or the asparagine of the calmodulin protein. Now, if you talk about the functions of the calmodulin, what happens is that when there is a high concentration, when the IP3 that is made, that is going to release the calcium from the endoplasmic reticulum into the cytoplasm. These calcium are then going to bind to the calmodulin, and if you look at the structure before the binding of the calcium, just for the sake of example, it looks like this. But when the calcium it binds over here, the conformation is going to change, and you can see a very clear change in the conformation over here. So when the calcium it binds to the calmodulin, this particular complex is known as the calcium calmodulin complex. And when the calcium calmodulin complex is made, then this particular complex it go and it binds to other protein, thereby activating them. So if you talk about the targets or the functions of this calcium calmodulin complex, they are very important in the regulation of cell cycle. They are very important in the intracellular signaling and in the muscle contraction. Now, if you talk about the importance of the calmodulin for a cell, so if there are mutations in the calmodulin, those mutations are actually lethal. In most of the cases, the mutation in the calmodulin usually proves to be lethal for that particular cell. Now, the allostatic activation of the calcium, of the calmodulin by calcium is analogous to the allostatic activation of the kinase A by the cyclic AMP. In the video on the protein kinase A, I've told you that the protein kinase A actually gets activated by the cyclic AMP. In this particular case, the calmodulin, it actually gets activated by the calcium. So this activation mode is almost similar for the protein kinase A and the calmodulin. But the difference between the two is that when the cyclic AMP, it activates the protein kinase A, the protein kinase A have got enzymatic activity. But when the calmodulin, it gets activated by the calcium, making the calcium calmodulin complex. So this calcium calmodulin complex has no enzymatic activity itself, but it can act by binding to other proteins, thereby activating them. Now, in some cases, this calmodulin, it can serve as a permanent regulatory subunit of an enzyme complex, but in most of the cases, the binding of the calcium, it enables calmodulin to bind and alter the activity of the other proteins. Now, if you talk about the one of the specific targets of this calcium calmodulin, so the special targets of the calcium calmodulin, that can be enzymes or they can be the membrane transport proteins. For example, in many cells, when the calcium calmodulin complex, it gets activated, it goes and it activates the plasma membrane calcium ATPase, and we had a detailed discussion on the function of the plasma membrane calcium ATPase, that it is actually responsible for pumping the calcium out of the cell if the concentration of calcium increases in the cytosol. So this plasma membrane calcium ATPase, when the calcium calmodulin, it binds to this plasma membrane calcium ATPase, this pump actually gets activated, and when there is a high level of the calcium in the cytosol, this pump actually help to return the cytosolic calcium level to normal by removing the excess calcium from the cytoplasm to the extracellular space. So this is the direct effect of the calcium calmodulin on this plasma membrane calcium ATPase. So this is the direct target for the calcium and calmodulin. However, most of the effect of the calcium calmodulin, they are indirect, and they are mediated by the calcium calmodulin dependent protein kinases. And the name is very clear that these protein kinases, their activity actually is dependent on the calcium calmodulin complex. So the calcium calmodulin complex can work directly by activating, like the plasma membrane calcium ATPase, but they can but most of their action are indirect, and they are mediated through this special class of the kinases. Now, when you talk about the calcium calmodulin dependent protein kinases, as I've told you, that these are the protein kinases, but their activity is dependent on the calcium calmodulin complex. This means that when the concentration of the calcium increases in the cell, it is going to make the calcium calmodulin complex, and this calcium calmodulin complex will then be responsible for the activation of these protein kinases. Now, these calcium calmodulin dependent protein kinases again are serine threonine kinases. What I mean by that is that when the calcium calmodulin dependent protein kinases, they act on their substrate, they are going to phosphorylate their serine or threonine residues. Now, these calcium calmodulin dependent protein kinases, for short, they are known as the CaM Kinases. So this is the calcium calmodulin dependent kinases. They are for short known as the CaM Kinases. And there are two types of the CaM Kinases. One is known as the specialized CaM Kinases. If I give you the one of the example of the specialized CaM kinase, that will be the myosin light chain kinase. And this myosin light chain kinase, what it do is that it go and phosphorylate the myosin in the smooth muscles, and when the myosin of the smooth muscle that is phosphorylated at the serine or threonine residues, it actually is responsible for the contraction of the muscles. Now, what are these smooth muscles? A little bit of definition of the smooth muscles. So along the skeletal and the cardiac muscles, one of the types of the muscles tissue in the body is the smooth muscle. and the smooth muscle generally form the supporting tissue of the blood vessels and hollow internal organs such as the stomach, intestine, and the bladder. So what you can see is that when the concentration of the calcium that increases in the cell, that is going to make the calcium calmodulin complex. This calcium calmodulin complex is then going to activate the myosin light chain kinase, which is actually a specialized CaM kinase, and this myosin light chain kinase will then phosphorylate the myosin at the serine or threonine residues, and when that is phosphorylated, that will be responsible for the contraction of the smooth muscles. The second class of the CaM kinases, they are known as the multifunctional CaM kinases. Now, as the name indicate, they have got multiple functions, therefore they are known as the multifunctional CaM kinases, and they are collectively called as the CaM Kinase-II. So when you look at this term 2 with the CaM kinase, that means that that particular kinase will actually belong to this family of the multifunctional CaM kinases. If you talk about the function of these multifunctional CaM kinases, they play important roles in the neurotransmitter secretion, the transcription factor regulation, and the glycogen metabolism. Now, I want to focus on the structure of this CaM Kinase-II. What is the structure of this CaM Kinase-II? So if you talk about the structural domains that are present in the CaM Kinase-II, so all of the isoforms in this particular family, in the multifunctional CaM kinase or the CaM Kinase-II family, all of the isoforms, they have got a catalytic domain, and the catalytic domain have got binding sites for ATP and substrate, just like the ATP and substrate binding site we just saw in the Protein Kinase C. So the CaM Kinase-II have this catalytic domain having binding site for ATP and the substrate protein, and this catalytic domain is actually responsible for the transfer of a phosphate group from ATP to the serine or threonine residues in its substrate. The second important component of the CaM Kinase-II, that is known as the autoinhibitory domain, and the function of the autoinhibitory domain is very much similar to the function of the pseudosubstrate that we just discussed in the Protein Kinase C. Now, what this autoinhibitory domain do is that it function as a pseudo-substrate site which binds to the catalytic domain. So this autoinhibitory domain, when it binds to the catalytic domain, that means that the kinase activity of the catalytic domain that will be blocked. Now, what happens is that this structural feature that governs this autoinhibition is the threonine 286 residue. What I mean by that is that in the autoinhibitory domain at position number 286 there is threonine, and when this threonine is in the dephosphorylated form, this threonine goes and it binds to the catalytic domain, thereby blocking its kinase activity. Now what happens is that if you phosphorylate this threonine 286, this particular phosphorylation will actually remove the autoinhibitory domain from the catalytic domain, and the kinase activity of the catalytic domain that will be restored. So the phosphorylation of the threonine 286 that will permanently activate the CaM Kinase-II, as this effectively blocks the autoinhibition. Now the other two domains that are present in the CaM Kinase-II, one is known as the variable domain and the other one is the self-association domain. Now differences in these domains that actually contribute to the various CaM Kinase-II isoforms. So in all of the isoforms of the CaM Kinase-II, you will find a catalytic domain, you will find an autoinhibitory domain, and there is a very much similarity between the catalytic and the autoinhibitory domain of the CaM Kinase-II isoforms. But these two domains, the variable and the self-association domain, actually they contribute to the variety of the CaM Kinase-II isoform. There are differences between the variable and self-association domain of different CaM Kinase-II isoforms. Now the self-association domain, it's actually found at the C terminus, and the function of this domain is the assembly of the single protein into a large multimers. So you can actually see large multimers between 8 to 14 subunits. So you may be having the CaM Kinase-II that may have 8 subunits. You may see CaM Kinase-II isoform having 10 subunits, and you may see CaM Kinase-II isoform having 14 subunits. And this multimer formation is actually because of this self-association domain. Now I want to focus on this particular thing in the next slide, and if you can see over here, this is the inactive CaM Kinase-II. And when this is inactive, you can see over here there is no binding of the calcium calmodulin. Now when the calcium calmodulin complex is formed because of the increase in the calcium concentration in the cytosol, the calcium calmodulin is then going to bind to the inactive CaM Kinase-II, thereby activating it. Now when the CaM Kinase-II it binds to the inactive CaM Kinase-II, it actually activates its phosphorylation ability. And when its phosphorylation activity that is activated, so one of the important site that it is going to phosphorylate is this Threonine 286. And when the autophosphorylation ability of the CaM Kinase-II after the binding of the calcium calmodulin complex it gets activated, it is going to bind to phosphorylate the Threonine 286. And when the Threonine 286 that is phosphorylated, the autoinhibitory domain will not be able to bind to the catalytic domain, and hence the catalytic activity or the kinase activity of the CaM Kinase-II that will get activated. And if you can see over here, once the autophosphorylation activity that gets activated, this actually then increases. And if you see all of the subunits of the CaM Kinase-II, then they get activated when they get phosphorylated. Now the best example of the CaM Kinase-II is the one that is especially enriched in the nervous system, and it is highly concentrated in synapses. Now if I give you an example of this, so when the neurons that use the catecholamine neurotransmitters, and in the family of the catechol neurotransmitters, the important examples are the dopamine, the noradrenaline, or the adrenaline. What I mean is, when the neuron that uses these neurotransmitters for their neurotransmission, when there is the influx of the calcium through the voltage gated calcium channel in the plasma membrane, it actually allows the cell to secrete their neurotransmitters. Now when this CaM Kinase-II, it actually need a calcium calmodulin complex. So when the neuron that uses the catecholamines and when their neurotransmitter release is activated through the influx of the calcium through the voltage gated calcium channel, this calcium influx also activate the CaM Kinase-II, because this calcium it binds with the calmodulin, the calcium calmodulin complex it then activate the CaM Kinase-II, and the CaM Kinase-II then go and phosphorylate an enzyme which is known as the tyrosine hydroxylase, which is the rate limiting enzyme in the catecholamine synthesis. So on one side, the release of the neurotransmitter or the release of these catecholamines from the neuron is depriving, or that is decreasing the concentration of the catecholamine in the neurons. So on the other side, the activation of this tyrosine hydroxylase by this CaM Kinase-II is then actually going for the resynthesis of the tyrosine, the resynthesis of the catecholamine, and the neurotransmitter they can actually utilize them as the neurotransmitters. Now the CaM Kinase-II that has a remarkable property. Now it functions as a molecular memory device. What I mean by that is, when it gets activated by the calcium calmodulin complex, it then remains active even after the calcium that is withdrawn. As I've told you that the activation of the CaM Kinase-II that is dependent on the calcium calmodulin complex. So when the calcium calmodulin complex it once activate the CaM Kinase-II, so if the concentration of the calcium drops down, the CaM Kinase-II still remains active, because it keeps the memory that the complex of the calcium calmodulin complex that has been formed. Now this memory, this molecular memory device function of the CaM Kinase-II is because the CaM Kinase-II has the autophosphorylation activity as well as other proteins. It can autophosphorylate itself. It can also phosphorylate other protein because of its kinase activity when it is activated by the calcium calmodulin. Now in its autophosphorylated state, the enzyme remains active in the absence of the calcium, thereby prolonging the activity of the kinase beyond the duration of initial activating calcium signal. For example, why are we having this molecular memory device property of the CaM Kinase-II? The reason is that we need the activity of the CaM Kinase-II for a long time, but we cannot keep the concentration of calcium high in the cell, because high concentration of the calcium is toxic to the cell. For example, if you need the activity of the CaM Kinase-II for 3 seconds, you cannot keep the concentration of the calcium high for 3 seconds. So what the calcium do is that when its concentration increases after 1 second, its concentration is taken back to the homeostasis level by different pumps that we discussed, by the Circa, by the PMC. But the CaM Kinase-II, because of its molecular memory device and autophosphorylation ability, is able to stay active for 3 seconds, and when it stays for 3 seconds, it can actually function properly. Now this when the activity of the CaM Kinase-II that has been carried out, the activity is then diminished by the action of the phosphatases, which actually removes the phosphate group from the activated CaM Kinase-II. And if you remove the phosphate groups from the CaM Kinase-II, the CaM Kinase-II is then going to be inactive again. Now, because of these properties, the CaM Kinase-II activation can serve as a memory trace of the prior calcium pulse, as I've just told you, and it seemed to play an important role in some types of the memory and learning in the vertebrate nervous system. For example, in the mutant mice that are missing the brain-specific subunits of the CaM Kinase-II, they were having defects in their ability to remember the location of an object that is known as the spatial learning. So the mutant mice that were missing the brain-specific CaM Kinase-II subunits, they were having problem in this spatial learning. So if you like the video, please subscribe to my channel, and hit the like button and share it with your friends.